Biology Teaching and Learning Videos. Semester 1. Homeostasis. Urine formation. 1. Ultrafiltration occurs at glomerulus and Bowman's capsule. 2. Reabsorption occurs at proximal and distal convoluted tubule, loop of Henle and collecting duct. 3. Secretion occurs at proximal and distal convoluted tubule. Ultrafiltration. 1. Glomerular filtration. Water and solutes smaller than proteins are forced through the capillary walls and pores of the glomerular capsule into the tubule. Reabsorption. 2. Tubular reabsorption. Water, glucose, amino acids, and needed ions are transported out of the filtrate into the tubule cells and then enter the capillary bloods. Secretion. 3. Tubular secretion. H+, K+, creatinine, and drugs are removed from the peritubular blood and secreted by the tubule cells into the filtrate. Proximal tubules. It starts from the neck of the Bowman's capsule, and it is highly convoluted. It lies in the renal cortex region. Proximal tubules. The wall consists of simple cuboidal epithelial cells that contain mitochondria and bearing a lot of microvilli on the surface. Microvilli provide large surface areas for movement of solute molecule into blood during reabsorption. Mitochondria will provide ATP for active transport of substance in reabsorption. Loop of Henle. It is a U-shaped segment of the nephron located in the renal medulla. It consists of two straight parallel limbs, into the renal medulla and an ascending limb. Descending limb. Thick segment, simple cuboidal epithelial cells. Thin segment, simple squamous epithelial. Has aquaporin channel. Ascending limb. Thin, simple squamous epithelial. Thick, simple cuboidal epithelial cells with many mitochondria. Distal tubules. It is greatly twisted like the proximal tubules and lies in the renal cortex. The terminal relatively short part of the distal tubule is called the collecting tubules. It opens into the collecting duct. The collecting ducts receive the collecting tubules of several nephrons. They pass into the renal medulla and join each other forming large ducts. These run through the medullary pyramids and open into the renal pelvis. Collecting duct. The location in the kidney where processed filtrate, called urine, is collected from the renal tubule. Presence of aquaporin channel depends on the presence of hormone, ADH. Ultrafiltration. Ultrafiltration is the movement of small molecules across the glomerular wall into Bowman's capsule as a result of blood pressure. Filtration occurs when high blood pressure forces fluid from the blood in the glomerulus into the lumen of Bowman's capsule. The glomerulus are permeable to water and small solutes, but not blood cells or large molecules, such as plasma proteins. The molecules that leave the glomerulus and the Bowman's capsule are called the glomerular filtrate. Ultrafiltration. The glomerular filtrate produced in the capsule contains almost the same as blood plasma composition except for blood cell and large molecules. Glomerular filtrates are water, nutrient, glucose, amino acid, vitamins, waste product, urea, ions, Na+, K+, Cl, HCO3, and some drugs. The adaptation to enhancing ultrafiltration. 1. High hydrostatic blood pressure in glomerulus. Due to the diameter of the afferent arteriole is larger than the efferent arteriole. Forces almost all the contents of the blood except large blood proteins, red and white blood cells, platelets and plasma protein, into the space of Bowman's capsule. 2. Presence of highly coiled glomerular capillaries that provide a large surface area for filtration. 3. High permeability of glomerular capillaries to the blood solute due to thin and porous filtration membrane. Reabsorption. The process in which useful substances are reabsorbed from the glomerular filtrate within the tubule and pass them back into the blood capillaries. Reabsorption occurs through passive and active transport. Occurs in proximal tubule, loop of Henle, distal tubule, and collecting duct. Reabsorption at proximal tubule. At the proximal tubule, almost 80% of the glomerular filtrate is reabsorbed. Glucose, amino acids, potassium ion, and other essential substances are also actively or passively transported from the filtrate to the interstitial fluid and then into the peritubular capillaries. Reabsorption at proximal tubule. Molecules reabsorbed. Buffer, HCO3, K+, passive transport. Nutrient, all glucose, amino acid and vitamins, active transport. Salts, sodium chloride, active transport. Water, osmosis. The adaptation of proximal tubule to enhance reabsorption. 1. The tubule wall is 1 cell thick. 2. The surface of epithelial cell of the tubule wall facing the lumen have numerous microvilli to increase the surface area for this process of selective reabsorption. 3. The epithelial cells of the tubule also contain numerous mitochondria that produce the ATP required to supply the energy for the active transport of sodium ions out of the proximal tubule. Reabsorption at loop of Henle. 1. At descending limb, molecule reabsorbed, water, has many aquaporin protein which act as water channel, only permeable to water but not to salt, sodium chloride and other solutes. Water diffuse out of the tubule by osmosis. Solute concentration inside the loop of Henle increases down the descending loop of Henle. Reabsorption at loop of Henle. 2. At ascending limb, consist of thin and thick segments. The walls is permeable to sodium chloride, but impermeable to water. At thin ascending limb, sodium chloride diffuse out by passive transport. This movement of salt adds to the high osmolarity of interstitial fluid in medulla. Reabsorption at loop of Henle. 3. At thick ascending limb, sodium chloride is actively transported as concentration of sodium chloride in tubule become lower. Filtrate becomes progressively more dilute as it moves up to the distal tubule. Reabsorption at distal tubule. 4. At distal tubule, substances reabsorb from tubule to peritubular capillaries. Sodium chloride, active transport. HCO3, active transport. Water, osmosis. Reabsorption at collecting duct. 5. Sodium chloride is actively reabsorbed because the collecting duct is not permeable to salt. In inner region of medulla, epithelium of the duct is permeable to urea. So, some urea diffuses out of the duct and into the interstitial fluid. More water is reabsorbed by osmosis and causes the urine become more concentrated. High osmolarity of the interstitial fluid in medulla contributed by sodium chloride and urea. The collecting duct carries the filtrate through the medulla to the renal pelvis and ureter. Secretion, a process in which wastes and excess substances are removed from the peritubular capillaries into the tubule of the nephron. Secreted substances include hydrogen ion, ammonia, many drugs, uric acid, creatinine and K+, transported by both active and passive transport. Occurs at proximal tubule and distal tubule. Secretion. At proximal tubule, H+ is actively secreted into the tubule to control pH level in body fluid. Ammonia is passively secreted into the tubule to act as buffer to trap H+ in the form of ammonium ions, NH4+, and prevent the filtrate become too acidic due to the presence of H+. Drugs and toxin that have been processed from liver actively transport into proximal convoluted tubule. Secretion. At distal tubule, K+ and H+ is actively secreted from the blood into the tubular filtrate to maintain homeostasis of electrolytes and pH. Therefore, there is a net loss of K+ in the urine.